Welcome everyone to today's version of the ReproNim webinar. We're pleased to have one of our own core members, JB Pauline, to present on topics about how next generation neuroinformatics tools should be developed. To the ReproNim crowd, JB needs no introduction. He's been working with the core ReproNim team since before there was a ReproNim, since there was INCF, since there was BIDS development and other things like that. He is old enough to be a core developer on things like SPM and other tools that are well known to the world. Practically speaking, he currently is part of the Ludmer Center for Neuroinformatics and Mental Health. He is a key influencer in the Canadian Open Neuroscience platform, and again, has a huge connections and leadership positions in the INCF, the International Neuroinformatics Coordinating Facility. All of these neuroinformatics ties make him a key cog in the ReproNim wheel to rule the world. So I'm sure there's many other things I could talk about. His CV is long, but if I've missed anything critical, I'm sure I'll let JB introduce anything else himself that he would like us to know. So thank you, and the floor is yours, JB.
Thanks, Dave, and no specific things to share. Let me share my screen, though. I think that would be good. Can you now see my screen?
Yes.
Yeah? Okay, cool. Yeah, so I wanted to talk a little bit of how a neuroinformatics project should be run or can be run. So there will be a little bit of that in my talk, and there will be also, of course, a little bit of the showing of what we've done, hopefully using some of those principles and recommendations and tips that I can share. The reason why I think working on infrastructures or data sharing aspect or those things, that I think it came to me because there's a bit of a tiredness sometimes of okay, this is the next paper, this is the next project, this is and I think finding ways of improving the general ecosystem rather than just one specific aspect of science in neuroimaging, for instance, was one of my motivation. Infrastructures are kind of knowing definitely huge work on Nitric and other things and ReproNim, infrastructures are interesting kind of objects in some sense. Like they're first of all, they are noticed when they don't work. That's one thing. Second, they're really intertwined with your scientific life. So if you're all using the web, when the web doesn't work, that's a huge disruption. And when they're built, they're really hard to change as well, because there's a long inertia on infrastructure aspects. So I want to having those facts a bit in mind before we embark in the journey of that. There's also aspect of a competitive space, infrastructures when there are, when you have a road or when you have some tracks, good luck trying to build another one next to it, because it's again long-term costly aspects of a project. So the reason still I think is important and critical to work on those things is that, first of all, I guess long-term data sharing aspect, and I'm talking about data infrastructure for data sharing and processing, I guess. Those long-tail data is still, I think, the #1 issue in neuroimaging or neuroimaging genetic world. The second thing is that reproducibility and generalizability crises are definitely not solved. We've been working on those things for 10 years or more, and we're still not getting entirely the success we hoped for. The analytical flexibility aspect, so just definition of that quickly, it's the fact that when you're using a package or another one, it's the sensitivity of the results that you get in biology or neuroimaging are maybe very those results may be very variable across the packages and those things. So that is yet not a blessing, and help us to understand and to build and to it's at the moment it still occurs. And I would say that governance of projects, and that's maybe one point that I think I would like people to be aware of. The governance of project is a major and often overlooked element in tool and platform development. And what kind of governance as those things is important. So the long tail in neuroscience, I think you all know that problem. The unpublished dark data is supposed to be 85% of the data that we see. Huge value worldwide, possibly. I think the question is not even whether those numbers are correct. The question is the thing that is undeniable is that the fact that there are a lot of lost data and dark data, and that makes like a small ends and all those problems of a sample size issue real problems. So one of the motivation for actually working on data sharing and what I'm going to present to you is really like the motivation of the machine learning motivation, that when you build a specific learner, let's say on a young population, and you want to know whether that population is prone to develop a disease or something, then how does that learner performs on another population, depending on how that learner was built, and so it's and often it doesn't perform well at all. So the problem is really to have enough data sets and enough population, enough cohorts, especially of possibly like non very represented population in your machine learning or in your learner, on your new deep learning system. And that's something that is not that easy to get in many instances. The other motivation is the analytical flexibility I was mentioning. This is just using 3 tools and looking at the correlation between brain structures across subjects, across those tools, and you see a huge difference between those tools. While those tools are all, well accepted, good tools that have been, used in many instances in many publications, they give you very different answers, and that's we still don't have yet a good way of actually just even a way of studying that problem is difficult, and therefore hence my kind of a push on the infrastructural aspects. So when we talk about project and and I think I guess develop neuroinformatics project in general, but I think there's really 2 key aspects. The sustainability aspect, the one is the usual problem that everyone has that in mind, or most of us have that in mind. How do I sustain my project for the future if I don't get the next grant, all those things. The governance aspect is a bit less, I think, prominent and seen, and I mean it has, but there's a number of publications, some of which actually participated into is to see what kind of governance make those projects more sustainable, and I think that is an important aspect of things. And so governance, I mean, the couple of principles: transparency, inclusivity, of course, open, accountability and trust. The no lock-in aspect, I think, is important as well. So let's say my project is not sustainable, is going to die, or because I don't have the resources or things like that. What is the impact on the people who have adopted some of those tools? So I think with for the sustainability and the governance aspect, the going distributed is definitely a way to go. Distributed funding model, distributed development, distributed and also based on the community standards and being lightweight. Like you don't build something that is too heavy because it's difficult to sustain and maintain. Implementation, there's a bunch of interesting aspects to mention. The portability, interoperability, the user-oriented aspect, I think are 3 things that came into my mind when I wrote those slides. So the one thing that I would say is critical, and one of the reasons why I'm so glad and grateful to be part of that of the ReproNim ecosystem and community is that there's the buzz factor for the development that we all know. So how many developers need to be killed to kill the project? But the problem is really the lab buzz factor. You know how many labs you need to are going to contribute and maintain and push one project is the thing that I think is important and critical. I don't want to be the buzz factor there. So so this is our some principles. So we built on standards and community of practices. We try to be cheap and sustainable because we are not we don't run a huge operation. We try to have distributed governance for distributed infrastructure. And work with the driver of the research, which, as Carl Goebel was saying, is love, fame, money, and rules. Hopefully love is the important one, but others are have a significant impact in motivation for in general. So on the project that I'm going to describe quickly, there are a number of things that we're doing. So definitely we rely on community and standards. We run the lean operation, and we are working on distributed governance for those projects. I think that's a critical aspect. So what are the two projects that we are trying to build on the using those principles? Well, they're NIPOP and Eurobagel, and I'm going to now describe those things a little bit for those who haven't heard about those projects. For those who have heard, I'm hoping that you can interrupt and ask me hard questions. I'm be always happy for that. So I guess the one aspect that has not been covered very well in the neuroimaging world is the flexible data sharing of local, possibly sensitive data. It's always difficult. The ethical and legal aspect, the GDPR in Europe, those all those even between Quebec and Ontario, I can tell you the laws are different for the data sharing, and it's not easy to actually move data and so on. So the flexible data sharing for data, that's one of the things that I really want, I think, to try to tackle a bit more. The reducing the barriers for entry to harmonize data sharing, and that's another thing. How do you make it really easy for folks that are not developers, not informaticians, and have little experience with even the terminal or those things? How do you make that feasible to get your data harmonized? And then how do you for those that are really working on the analytical pipelines and things, how do you make that a flexible framework for studying analytical flexibility? And then the governance aspect should be embedded in those things. So I'm going to switch to those two projects, and I'll be hopefully quick, and so that we can have time for discussions. And if I can make that happen. I subtitled Decentralized Neuroinformatics Framework, Standing of the Children Giants. And of those giants, I want to definitely mention the IDM framework, the DataLab project, all the ReproNim tools, and the bits that standards, and the bits derivative standards, and all those are the giants on the top of which those projects were built. And I think that's very important to recognize that that genealogy of those two projects. So on the left, there's a bunch of people that have worked on those projects. There are actually quite a few more now. This slide is already old, and the distributed development aspect is really starting these days. I mean, have been started recently, and I'm so happy for that. So what's the end goal of that framework is to really simplify the problem of having data at different places that are not harmonized and that are, and how do you find them and how do you reuse them? How do you make them fair? Using, solutions that exist already, but also using very specific use case so that we don't get lost in the kind of, okay, we're going to solve all the problems. And maybe others can do that. I'm not able to do that. I want to try to see one problem and try to solve for that problem. So two projects. NiPoppy is a kind of a specification for processing aspect. NeuroBigel is for sharing across institutes. So what is the challenges? What are the challenges? Curation to head sites at the bits. So there's a lot of when I said building on the top of on the shoulder of giants, there's the UDECONF is one of those giants over there. Processing consistent pipeline execution aspects with data lab, of course, data lab and other things. Tracking is really important as well. I think we have not get that part very well in other things, so that's another aspect, and that's a work that Michel and Nikhil and the lab have started, and Brent as well. And I'm so grateful for it's been pushed in a very good place now. And then there's the harmonization problem. How do you harmonize things, align across data sets, and discover across those data sets that may not lie in the same infrastructure. So NiPoppy is an organization and decentralized processing of neuroimaging clinical studies. It's really a specification rather than something else. It's like if I have to describe it in one word, that's more like a specification than anything else. For not only imaging data, and that's the critical thing to me, is that there are some tools that are there are many tools actually, or quite good tools to think that are dedicated to the imaging aspects. There's much less. I think. I mean, of course, with bits, phenotypic variables, and all those things, there are things, but there are much less. Things that integrate non-imaging data in the same framework. It's also a tool for a protocol for how do you go from A to Z, when you've got your raw data to your curated and then processed data. So that's an aspect that I think is important. So those are specification, protocol, and then it comes with a set of tools. It's an ecosystem in some ways, and set of tools. It's not like a monolithic aspect. It's a very kind of flexible specification, and maybe like a bit of a playground for like a standardization of a whole study processing. So why? Because the messiness, and it requires a lot of flexibility. The problem of curation organization is really time consuming, error prone. There's a lot of existing tools, so let's build on the top of them. And in terms of the landscape, you've got a lot of a centralized platform. I would say maybe BrainLive, CBrain, all those things exist and they are really useful. They just require that data are uploaded sometimes, and it's hard sometimes to go from one to the other. So the interoperability is not always great. There are some decentralized platforms and really good ones. They often have a set of and are not very flexible. So they also sometimes don't follow community standards. And there's also plenty of other things that exist. And in that landscape, often we found that there's a limited capacity of handling non-imaging data for those platforms. So that's those are the reason and the rationale for developing that ecosystem, which is I think more flexible and not monolithic. So what does it do? So you take your input data. They could be the DICOM, the NIFTY, the REDCap, the Excel spreadsheet aspect or CSV kind of thing. Then you organize those things. We extend and reuse the bits phenotypic data protocol standards and the imaging derivative standards. And then and we can reproduce any pipeline. We use the framework of the of Boutique at the moment. And I think it's a really good framework. So you can get the provenance information from there and then assess the availability, completeness of those things with a set of, that's an idea of the NIPOPI framework, is that you have a set of functions that actually look at the disk and say, hey, you've got those files on that disk. That means that your FreeSurfer or FSL has run properly. And you got, and then you can extract automatically those sanity check of, beyond just the log of your Slurm or the log of your HG, like your QSUB or whatever. So once you have done that and you have like that standard, it's easy to actually create a set of tools to look at, what has happened to that data set specifically. And then you can, and this is a very lightweight web tool to actually look at your data sets, what has been successful in terms of processing for FreeSurfer, fMRI prep, like MRI QC, all those things. And then you're looking at those, and then you can, filter those things. Hey, how many subjects you have that actually run, FreeSurfer and also have a 2 session or something like that. So the capacity to actually interrogate quickly, a data set that is being processed and that has new subject coming in, all those things. That's one thing that we can provide with those tools. So a typical NIPOPI protocol, you initialize an empty data set, you add some configuration files, you download or you copy unzip your raw DICOM, you organize those DICOMs. They often need that step, and that's again a little bit of a training aspect in that specification, right? Then you convert to bits, then you process through those containers, and then you track the processing status, and then eventually extract image-derived phenotypes of interest locally. So that's a whole again, and everything is kind of lightweight. Everything is sort of in a CSV files. There's no huge demand on the IT aspect of for that specification. So that's was one of the design principles to it. So it combines a number of framework, standardized organization, flexible reproducible pipelines, tracking and IDP, tracking of output and IDP extraction, building support for ongoing studies. I think it was one of the things for longitudinal aspect as well, and it's a fully decentralized setup. So hopefully, we hope that this becomes a specification that can be retaken and developed by others. So once you have that, you can already ask questions such that, say, how many PD patients do you have that are under 65 and 2 imaging sessions, whatever, and you can ask that locally across data sets using those tools. But then when you have another data set, you need to need another tool to actually look at your cross data set, what is happening. And that's where harmonization and query across things happen. And that's even more important is that another colleague of you in another hospital, another neuroimaging center or a research center has the same sort of output or data, and you would like to know if the findability and the accessibility of those data are important. So the question is, how do you harmonize across institute and things? And we all know that everyone has their own way of doing things. So the way we approach that is to actually create a very simple, very down-to-earth tool to actually say, hey, if you have a column that is named diagnosis, and this is a DX is maybe is the column, and there's patient or there's Parkinson or patient or control or whatever, and then another institute would encode things in a different way. Then we need a common vocabulary. That common vocabulary, what we do is we embed in a web tool to actually say, hey, you've got your CSV file, you've got your Excel spreadsheet. Let's paint and say that age is age, that diagnosis is this column name. So it's a painting thing, and then once you have painted the column, say, hey, this is age or this is sex, then you can go within that column and say, hey, how many unique variables do you have in that column, and then you or are missing or things like that, and then you can look at and using for instance for the diagnosis aspect, we're using OMUP SNOMED CT to actually tag those specific cells, not only the columns of that Excel spreadsheet, but the cells. So at the individual level, we have this information. So that's a very kind of a easy to use tool and easy to adapt to another kind of a model. And what it does really very little. It takes the TSV file of a participant.tsv file of and add some information that are machine readable. The TSV file that is the description JSON file that is in the bits data set is for humans. So you have a description edge of participant. What you do is you actually add that little annotation that is machine readable for it. So it's a very lightweight thing. And if everything falls down and burns down, the thing that you still have is that data dictionary that is machine readable aspect. So once you have that annotation for each of those data sets that are distributed in different institutes, then there is the interesting aspect that you can query across those harmonized. So you can spin off like a little bit of a graph store locally, and then create a query for querying those data across institutes. And the problem here is that querying, not everyone can write Sparkle. I can't, although I would like to. I mean, I've done a couple of Sparkle queries in my life, but not it's not a job for the research scientist or the so we created this kind of a tool that does the interface between the human query to to a machine query. And today we're already federating across several nodes, like OpenEuro is one of them, and those nodes will respond with on those queries. So let's say I'm looking for Parkinson's disease and female, or that have 2 sessions and T1 images and all those things. And then what the thing answers, I don't know if you see on my screen, answers the number of subjects you have found in a specific data set. So it's not answering the data set, it's answering the actual lines and the actual rows of those Excel spreadsheets. Those are the subjects that match your query. And then we have when there are data sets, we have the capacity to say, hey, this is where the file that you're interested in is in the using data.gitanex framework. So this is what we've done, and the important aspect is that locally you will be able to control. Can I actually say? Can I give all those results, all those age and sex and data that are associated with the subject? Maybe the UDPRS, the MoCA values, all those things. Can I do that, or can I just answer that yes, there are data, or can I answer yes, we have 70 subjects or 51 subjects on that answer your query? But that's not. But I can't tell you more until you sign this the data usage agreement, or you sign this consortium agreement and so on. So it's a way of keeping the governance local and flexible, and that's very appealing to a lot of people, because that's a real need in many clinical data sets, for instance. So this is what has been built, and I think now we're starting to have a lot of interest in that thing. Again, thanks to building on top of all the previous ReproNim tools, and I personally think that is already a ReproNim tool, and I think that's those control access, but local control access that you can tune quickly and easily are really critical to make data sharing much more widespread than it is at the moment. And you can, when you have those nodes, you can, everyone can see the open nodes, but not everyone. I mean, so we have some nodes that can't actually show at all who they are and what they're doing, and on the network. There are maybe psychiatric hospitals. We have 2 psychiatric hospitals that have nodes. You won't see their data, but they will see, the data that are available either on the OpenEuro or Indie nodes that are, open, have CC0 licenses or things like that. And we're building that across, in Montreal with different partners, but also across the world for with the Parkinson Parkinson use case in mind to first. So like, with Stefan Loesi, with the iceberg cohort, with the Shweta Parad in Nimans, with their cohort over there, with Enigma PD, with and so on. So that's where we're at. And actually on Open Neuro itself, if you look at searchable dataset, there's a little link that you might see, like search participant level with NeuroBagel, that you can actually launch those search. It will actually search across not only Open Neuro but also Indy and QPN and soon other institute or data repositories. Future development, there's a lot to be done on many fronts. One thing that we're working hard on now is the authorization and authentication of authorization aspect. Again, to make sure that there's an easy possibility of sharing with some colleagues that that have signed those things and facilitating that sharing when a larger sharing is not possible. And we're thinking like, studying and looking at the Global Alliance, the GF4GH, the Global Alliance for Genomic and Health tools that have this concept of passport and visa. Passport to tell who you are, and visa to say what do you have access to. And we're building that with a number of institutes, again, in the spirit of distributed development and adoption. We're also looking at various large language model to help the process, especially the process of harmonizing data locally. I think that's one thing. Possibly the query process as well. I think Jeff Grether at UCSD has done some work already on that, and in the institute and I think that's those things would be useful to add to the ecosystem. So please visit the website, and I thank you very much, and I hope to have a good discussion on that.
Thank you, JB. I guess I'll ask an initial question. It's not going to be that hard of a question. I had a gut reaction to one of your early claims of analytical flexibility is a curse, not a blessing. I wanted to know that if we were to fix sort of these, I sort of agree with you or I do agree with you when the infrastructure is the chaos that it is currently. Is it true that I can hope analytical flexibility can be welcomed back into the fold once we get this infrastructure and provenance and tracking better taken care of, so that then we don't have to rely on one particular sort of standard way of doing things, and we can let the 1,000 flowers bloom and learn about the best ways to handle things in sort of that analytic flexibility mode? So is the type of things that you were promoting, and we ReproNim promote as well, a path out of the analytical flexibility curse, or am I fooling myself?
No,
Sorry for the long question.
I think that having the tools to study analytical flexibility is still not easy thing to have in the kind of a standard neuroimaging lab. I think that's statement one. It's you go to a random lab, they use FSL to do X, they use FreeSuffer to do Y, they use and once you've got your results, you've processed those 75 subjects, participant data. Why on earth would you spend another 2 or 3 weeks or and headaches to actually run that with another pipeline that does about the same thing, that has the same credentials in terms of in the literature and all those things. And the reason is that it's just painful to learn those tools. It's difficult to so all those framework that helps running several times and several things and study analytical flexibility are welcome. So the BAPS is a welcome project. BrainLife is a welcome project. All those Neurodesk is a fantastic project. All those things are a great project for at least having capacity to do that. I think we don't have yet a good set of tools and methods to gather all those results that are coming from different pipelines, possibly weight them in some ways and say, hey, okay, now that's the consensus, or now that's, so we have crude ways. I mean, for instance, in one of our study on essential tremor, what we say, hey, we're going to say that result is valid if only if it can replicate across pipelines. So we're going to use free pipelines, and if we don't get the same results with the free pipelines, we will say that that's a safe, or enough safe to say that that we get those results. So I think that's one thing, that we don't have the tools to do that. One aspect that will be interesting is that for learning aspects. So let's say you want to learn diagnosis, or something from those image-derived phenotypes, and you, instead of having one set of image-derived phenotypes from a specific pipeline, you have free set of image-derived phenotypes, and maybe that will help, because then machine can learn how to weight those different image-derived phenotypes to actually learn something. So in that framework of machine learning, is interesting to have. I mean, that potentially that's a really good framework to actually say that's going to be useful to have more than one pipeline, because I can take the information from different pipelines on various aspects that they are tuned for, and learn some diagnosis, something to predict on a specific subject. So I think that's but I don't think I've seen many. Maybe some of us have, but I haven't seen yet many of those kind of studies showing that it's actually useful to process with different pipelines to get about the same information. And maybe again, if someone has, I'd be happy to see. I'm sure it will happen, or it probably has happened. I haven't seen it, but it's yeah. It's not. Let's say it's not common yet, for sure.
Questions from others? Don't be shy.
Following up on what Dave was talking about, the idea of analytical flexibility kind of reminds me of Chamberlain's concept of the theory of multiple hypotheses, when coming up with an explanation for the data, or for coming up with an experiment, where except in this case we have the same data and we have multiple tools for analyzing the data. We almost can't be biased, I guess, in the tool that we used, or how do we prevent bias in the tool that we use from driving the result that we I don't know, maybe want to publish or desire to publish.
Yeah, it's there's a p hacking problem that could be the pipeline hacking problem.
Yeah.
So that's true. I think there's no. I mean, to me, the solution to that problem is at the literature, the reviews, and the editorial task level. Reviewers and editors should start to ask, hey, you obtain your results with FSL 612. Why don't you try with another pipeline, and can you show us that you have it puts a huge burden on researchers, and it's not an easy thing to ask. I guess the community would say, hey, but that has been validated 1,000 times. Why do you ask me to do it again? So we have to we show that we get different results, and therefore it's worth. And but it's not something that anyone wants to do on the side of producing results, because it's a pain. And it's unclear how do you then describe the results that are maybe significant with one pipeline and not with another, and it creates headaches and difficulties that are not. There's no clear recipe or like a framework to deal with those things easily. So I think it should be still required, because that's that. And like it does, I think analytical flexibility for sure has a massive impact on the reproducibility crisis that we see. So I think it is still useful and commendable to do. And I'm just not seeing it very quickly happening because of the practical aspects of running those things. And interpreting both of the on the running and the interpretation aspects. Yeah, right. You must have a question, unless you.
All clear. How better we could work with standards? How we could make standards even more prevalent? How could we make those boutiques to be bits app to O apps, just those little things,
Yes, I mean, those are not little things. But yeah, no, I think it's I think making sure that those things reach the standard level and the and community reviews and those things. I think for NIPOPI specifically, having a playground of on how to do that and see how that works, and I think is a useful is useful anyways. Hopefully there's be sufficient community pushback to say, hey, NIPOPI, you should do it this way, like what you told us sometimes, and then we change things, we make it more more reasonable in terms of standards, and I think that's to me that's the goal, that makes make a playground for standardization aspects. Again, not only on neuroimaging, because I think that's the limitation that we are a little bit siloed in our community about. So I think that's where things we should push to other frontiers. All the clinical phenotypic data, of course, but also genomic stuff.
Just a sign of warning: you're going to OHBM hackathon, and your people, NIPOPI people, going to OHBM hackathon, so it will be a playground there.
It will be a playground.
We'll take some data sets and NIPOPI them left and right. Yeah,
exactly.
Other questions from the crew? I guess I'll just throw one out also as well. Again, the things that caught my ear from your original set of claims. I understand the topic of sustainability, particularly when it comes to tool development and data repository development and things like that. And when it comes to the more regular R01s or research grants that do sort of biological topics, yes, people would love to have the R01 that lasts 30 years or whatever and follows some similar topic through that, but that isn't reality. Most R01s are 3 to 5 years and done, and the investigator goes off in another direction. So there's sort of a non-sustainability built into so much. I mean, there's not an intention for sustainability built into most current research projects. It's the sustainabilities get your publications, so you can write some tangent grant and not necessarily sustain the same thing. So to some extent, we need to build non-sustainability, or the fact that most things do die is a reality, and very few things get sustained very long term. And again, there's a handful of infrastructures that do, but most research doesn't. Is there anything about building for unsustainability or proper end of life? And maybe to some extent, that's what the data sharing mandates are trying to do better with one of the mill R01s, is make sure that all that data lives beyond, has a sustainability beyond the funding. But anyway, I just wanted to probe more about sustainability in the context of, the sort of the biological research side of things, in addition to the sort of technology infrastructure side of things.
Yeah. Sorry, I got a call. I need to go. Yes, so I think it's to me the goal would be more that yes, the project will die at some stage, which is going to be replaced by something else and things. It is still going to be more useful and more long term, and therefore more worth investing in if it's a project across many labs, if it's a project across a large community. And that's it's harder, it's slower, it's and BIDS knows that very well. There's all those discussions and sometimes heated discussion, all those things. Like it takes it's not easy to progress quickly with with a large community and a lot of inputs and and a governance aspect that is distributed and all those things. It is still more long term, yeah, right. It's still going to take if for instance I often take the example of nylon. Nylon has 5-6 champion labs, and when there's no then one lab doesn't have the grant for paying a maintainer, then another one may have. And so there's this redundancy aspect, this that is useful. Like BIDS, for instance, there are maintainers of BIDS in so many labs now, or at least quite a few, and so it's not it really helps to have that community aspect for the sustainability and making sure that things are not too short-lived. I think that's yeah.
Great. I know you have to run. There's no last question keeping you from your student activities. Hearing none, I will thank you for the presentation today, and announce that, of course, next month we will have someone from Yarik's team. I think Christian Horia, perhaps, but someone from the Yarik team will be presenting next month. See you all then, and thanks for joining us today.
Take care.